So hello, everyone, and welcome back to our µTAS 2021 YouTube Live series. Today, we're featuring to be presented at µTAS research teasers from around the world, with Europe and Africa this morning. And now we have research from the Americas. I'm excited to introduce our very first guest, Professor Fu from the University of Michigan in Ann Arbor.
Thanks, Ryan. I'm going to go ahead and share my screen. And all right, so do you see my screen, right? Slideshow from beginning. Okay, all right. So hi, µTAS community here. And so it's great to be back. My name is Jianping Fu and a faculty member at the University of Michigan, Ann Arbor. So today I would like to give you a very quick preview about one very exciting project, or one project I'm very excited about in the µTAS 2021. So we're going to present an ongoing research project, building human central nervous system using microfluidics. The title is somewhat big, but it serves its purpose. All right, so in fact, in the past, over the last 10 years, my group have been using human stem cells, trying to develop different models, allow us to understand early human development. So in fact, over the last few years, we have been publishing a few visible papers and demonstrating that human pluripotent stem cells in suitable culture environments, they can self-organize, develop into multicellular structures, and the development of such multicellular structures mimicking early post-implantation development. So you can see that I'm going to replay the top video here. Let's just watch the video here. For the sake of time, we are not going into details, but let's watch what the cells can do in a 3D conducted environment. They can self-organize, form a cavity, and they can exhibit features associated with spontaneous symmetry breaking. So you can see that the cells organize form cavity. At the same time, a portion of the cells in the cell colony, they differentiate, they become somewhat different from the rest of the cells. So this is what we call embryonic asymmetry, symmetry breaking, mimicking some critical events in post-implantation human development. So if you watch the second video here, so you can see that the cells somehow, this cluster of cells, they were doing the same thing, open a cavity in the center where the cells, some portion of the cells in the cell colony, if you follow my mouse, they will start to differentiate as this pole, where the remaining cells remain columnar. But soon after, the cells start to disseminate, migrate away, and they are exhibiting what we call gastrulation-like events. But nonetheless, such three-dimensional culture environment and are not necessarily very controllable, repeatable. In fact, the efficiency for us to see such very interesting symmetry breaking events or gastrulation-like events, again, these are very important embryonic developmental events associated with post-implantation human development. The efficiency is somewhat low. Only 5 to 10% of the cell colonies can exhibit such interesting behaviors. So over the last few years, we reported, in fact, leveraging our background and expertise in microfluidics, we can make the development of such embryo-like structures, their development much more controllable and robust. So for example, if you follow the top video here, I'm going to replay the video here. You can see that this is a video showing that synchronized development of 9 cell colonies. They're exhibiting the same behaviors in a very controllable way. So if you follow the video very carefully, you see that I'm going to replay the video here. You can see that the cells will self-organize. These are embryonic stem cells, human stem cells. And soon after, you can see that they are forming a cavity in the center. If you watch very carefully, the cells at the top part of the cell colony, they look somewhat slightly different. Well, at the same time, soon after, the cells start to, from the bottom half of the colonies, they start to migrate away, exhibit what we call gastrulation-like events, mimicking human gastrulation. And you can see that these are the staining images showing the uniformity and reproducibility of the staining results. So these are what we have been published over the last few years on early post-implantation development. But in the same time, my lab is also very interested in developing model systems to model human central nervous system development. So we published a few papers in the past. And for example, if you understand human nervous system development, in fact, human nervous system development starts after gastrulation. And after gastrulation, when the ectoderm is there, and the ectoderm will self-pattern into what we call a multicellular tissue containing the neural plate. The neural plate is the precursor for the central nervous system. The neural plate will fold and fuse into the neural tube. Well, of course, the neural tube will continuously develop eventually form the forebrain, middle brain, hindbrain, and the spinal cord. This is the entire central nervous system. In the past, we have been publishing a few papers modeling different aspects of this human central nervous system development. But nonetheless, in this µTAS, I'm going to introduce an unpublished work. We're going to really demonstrate that using microfluidics, we can really show you that we can use human embryonic stem cells in a microfluidic environment to generate a completely patterned human neural tube-like structure. I'm going to show you data. In fact, I'm going to show you data. For example, you can see this is a slide containing some data. I'm going to show you some data, show you that we have a neural tube-like structure, a tubular structure, and somehow using microfluidics, we can pattern different regions of the tube with different identities, cell fate, mimicking early central nervous system development. In fact, we can also collect the cells for more careful downstream genomic analysis and compare our transcriptome data with existing in vivo data from either mouse or maybe even primate systems, and to really identify, validate the cell identities generated in the microfluidic system. So with that, I think it's time for me to close my talk, and I look forward to seeing all of you at µTAS 2021. And thank you very much. Ryan?
Wow, well, that's incredible work. And so I personally definitely look forward to seeing a lot more of that at µTAS in Palm Springs in the fall. And so thank you again. And then, so next, I'd love to introduce, we have Professor Sikes from MIT. And so Professor Sikes.
Yes, thank you for giving me the opportunity to participate in this event. And I would like to talk about some work on microfluidic paper analytical devices. And I've been excited for many years about the possibility of this kind of micropad joining the more traditional lateral flow tests that we're all familiar with in our global arsenal of tools to make measurements outside of laboratories. So the simplicity of paper and ink that was introduced to us and developed by George Whitesides and a number of his very talented researchers who have gone on to start their own labs, Scott Phillips, Andres Martinez, and Charlie Mace, really introduced, captured a lot of people's imaginations, including mine. And in the intervening years, it's been hard to bridge the lab to market gap. And it's also, if we're all very blunt about it, it's been hard to make the micropads, microfluidic paper analytical devices perform as well as the lateral flow tests. It's been around since the 1980s. But it's a very successful and easy product line, and it's hard to beat. It is more complex than just having paper and ink. If we look here, there are a number of materials that are encased in this plastic cassette, and they all have to be aligned with robotic pick and place manufacturing processes that during the COVID pandemic, they've been scaled to impressive levels. But it's been slow, as we've all seen. It took many months for this kind of test format to hit the market. So with my lab and a really fun cast of collaborators over the years, I've been working on new technologies that may help bridge these gaps, that may help us be able to scale the micropad devices, and then also increase their performance up to the standards set by lateral flow tests. And so in a lateral flow test, typically you have some porous membrane material. There's a capture antibody that binds specifically the biomarker that is in blood or urine or saliva or sweat or really any bodily fluid. And then once it's been captured and concentrated in a test line in the membrane, there's another antibody that carries some label with it that allows the user to tell if the biomarker was present in a yes/no kind of readout. And so as engineers, we've been working on many parts of the system. We really view it as a system where all of the parts can be designed and need to work together. So we've changed the biomolecules, the capture and the reporter reagents in this test format, taking inspiration from nature. We clone genes from thermophilic organisms and then use protein engineering techniques to rapidly tailor them to the use that we want to capture and label any disease biomarker. We've also changed the reporting chemistry, how a user determines whether the marker was present or not, rather than using particles or enzymes in some cases to generate color or electrical signals or luminescent readouts. We've been using chemistry, using autocatalytic reactions that lead to polymerization events. So you go from a liquid solution to a hydrogel or a solid. There's some kind of phase change that allows the reader to get a yes, no out of the device. And then also we use engineering design rather than treating these systems as a legacy format that needs to be extensively optimized for every new use. We take what we know about binding kinetics and transport, about convective and diffusive flow, and try to really make these into engineered systems rather than trial and error with 20 to 50 variables. So after working for many years on parts, we had the chance this year to put all of the technologies into tests and do real world benchmarking against every other diagnostic developer in the world in real time. So we got our courage up and jumped into the RADx Shark Tank, which was the United States experiment in accelerating diagnostic technologies. And we weren't gobbled up by the sharks. Instead, we had the chance to work with scientists and engineers at 3M to make µTAS, to take them out of the lab and see what are the complexities involved in manufacturing them at pilot scale and benchmarking the resulting performance. So at µTAS, I can talk about what we found, about the experience, and we made a number of tests. We made tests that diagnose infection, tests for the virus. We made tests for antibodies, any antibodies, binding antibodies, and we made tests for neutralizing antibodies to help one know if they now have immunity due to natural infection or due to the vaccines. And it's also possible in this format to test for neutralizing antibodies, immunity against all of the variants as they appear. And so it's a interesting story. I definitely learned more in the past year than in maybe the 5 or 8 years before it. I'm eager to share it, and really eager after 2 long winters in New England to soak up some of the California sunshine and get together with friends and fellow microscale analytical enthusiasts to chat about all the things that we tried in the lab that didn't work, that don't make it into papers, and just really enjoy the community of scientists and engineers who are excited about all of the same topics.
Wow, so that's great, and certainly impactful work that I think is gonna be really great to hear about more at the conference. And maybe some people will be using some of the devices you made to be able to enter the conference. We'll see. All right, so last but not least, we have Professor Carlo from the University of California, Los Angeles.
Yeah, thanks so much, Ryan. It's a pleasure to speak to you all today. And I am from California, so I've been soaking up all the sun even in the midst of this pandemic. But yeah, so I'm a professor of bioengineering and vice chair of the graduate program in bioengineering at UCLA. And when we were talking before we all started speaking, and I was remembering my first µTAS was in over 20 years ago, and in 2000 in Monterey. And I remember packing bags and putting books into the bag for all the attendees to have their conference proceedings. But certainly things have changed a lot since then. So, some of the recent developments from our lab that I'm happy to share, and these are new directions that we've been going. In particular, this idea of lab on a particle technologies. And so the idea is that we're using microfluidics to create small particles, less than 100 microns, between 40 to 60 microns. Typically, these particles have cavities so that they can be similar to, like, you might think of a well plate where you can have cells settle in them. In our case, we can seed cells, the cells adhere in the cavities. But these new particles, we call them nanovials that have cells within them. They now are able to be emulsified, so you can kind of separate each of these compartments, just like you might separate the wells on a well plate. You can have reactions performed in those compartments, or have secretions from the cells accumulate in those compartments. For example, we've looked at assays where we have antibody secreting cells. They're captured in these nanovials. They secrete their antibody, which we can capture similar to doing a ELISA on a well plate. In this case, we then have a secondary antibody that binds to the secreted antibody to label it. And so we get fluorescence signal associated with the secreting cell. And now we can do this massively in scale, right? So these lab on a particle technologies are also compatible with flow cytometers and fluorescence activated cell sorters. So you could sort these individual cells now based on their secreted products at scales of hundreds of thousands of reactions. So compared to well plates, where you might, 96 well plates or 384 well plates, we're now scaling orders of magnitude more. And we can do measurements on individual cells because the volume for each of these nanovials is thousands of times smaller than the well of a well plate. The advantage beyond using droplet microfluidics for these types of applications is, one, the end user doesn't have to have an expertise in microfluidics. Two, you can perform assays on solid phase. So in a droplet, it's hard to do solid phase reactions. So you can do washing steps and the traditional steps you do for sandwich assays. And then finally, you can analyze them using standard equipment like flow cytometers. So this is an exciting platform that we've started developing, and then we'll share more of our recent results at µTAS on applying this to a number of different applications where secreted products of cells are important. But I would also present kind of a new platform, which we call pico-shells. So the previous approach that I mentioned is we call them nanovials because there's a cavity, there's an opening. So the end user can load cells in just like a well of a well plate. Pico-shells is slightly different in that we use microfluidics to create a completely sealed cavity. So in this case, you're looking at a microalgae that's in one of these pico-shells. There's a hydrogel barrier around the shell here that's permeable. So unlike a droplet, you actually have media on the outside that can freely exchange into the cavity of the pico-shell. And that allows these cells to grow rapidly and over long periods of time. And then these pico-shells can be sorted using standard flow cytometers and fluorescence activated cell sorters because, again, they're all in a aqueous phase. So there's cell media on the outside, there's cell media on the inside. And there's cells that can be growing in these compartments. So we're applying this technology to a number of areas where biomanufacturing is important, where you want to select cells that are producing some bioproduct, whether it's internal to the cell, like lipids and microalgae, or it could be secreted products. But the challenge with this field in the past has been you can select cells that accumulate a lot of lipid, but they end up not growing much. And so here you want to select based on the combination of growth, biomass accumulation for lipid in one assay. So basically the way you can think about this is each of these pico-shells is one clonal colony that we can isolate we can still grow it in production relevant environments, and then we can do selection processes on it using standard instruments at scale, so hundreds of thousands. So I'm really excited to attend and see folks at µTAS. You know, one of the, beyond the amazing scientific discussions and you know, off the cuff technical discussions, and a lot of that happens at the conference, but a lot of it also happens at you know, venues around the conference where you have really great food. So here, this is my postdoc advisor, Mehmet Toner. We were having an amazing dinner in South Korea in 2015 µTAS. And then this is a few years back in Switzerland, where we had some of my group and other members where we're having fondue and talking science at the same time. So good food, good company. What more can you ask for? Looking forward to seeing you in this fall. Thanks.
Great. All right. Well, I agree with all of those sentiments. And so I just want to thank our guests again for sharing their work, especially their unpublished work with us today. And I invite everyone watching to tune in later tonight when we'll have our final set of research teasers from Asia and Oceania. And so thank you again. And we look forward to seeing you hopefully at the conference. Bye, everyone.